Welcome to 2.5.3, and this is, I didn't put this on here, but this is 2.5.3, protein synthesis. So 2.5.3, protein synthesis. And protein synthesis is called the central dogma to molecular biology. This is what everything else in molecular biology is based around. That we have in our nucleus this DNA molecule. And within this DNA molecule, we have a gene. Some segment of that DNA that it's going to code for a protein. And we're going to take that segment and we're going to transcribe it into what's called messenger RNA. And that messenger RNA is going to leave the nucleus, go into the cytoplasm, find a ribosome, and create a polypeptide, otherwise known as a protein. So this is the central dogma to biology, protein synthesis. So there's two parts, first being transcription. And transcription is going to be taking that gene and making, going from DNA to messenger RNA. Transcription takes place in the nucleus, and so here we can see that we've got our DNA molecule and we've got our messenger RNA coming off of it. So this piece right here, I'll use the line that they have, is messenger RNA. And then here, I'm not going to use that line anymore, but this is our DNA molecule. We're inside the nucleus, and this gold ball right here is the only enzyme that we are going to use. The only enzyme we are going to use is called RNA polymerase. And so if you remember back to replication, polymerase was our builder. And you can see that RNA polymerase is building a strand of RNA. Remember from our talk in class about RNA, uracil is taking the place of thymine. Also messenger RNA, or all RNA, is single-stranded, unlike DNA, which is double-stranded. So we're going to transcribe a piece of DNA into messenger RNA. So here's transcription again. We have our RNA polymerase, this big molecule here, and really it does the work of several enzymes that we had in replication. It's going to open up this bubble here just big enough so that we can bring in RNA nucleotides to match up with our DNA bases. So you see here that we've got a DNA base sequence of T, A, G, G, T, T, A, and we're bringing in our RNA bases C, A, U, C, C, A, U, and so on and so forth. So RNA polymerase doesn't only build, it unzips the DNA molecule, it builds, and it allows it to close back up, the DNA molecule to close back up. Alright, so RNA polymerase really acts as helicase and a polymerase. And you'll notice that our RNA strand is building from 5' to 3', just as our DNA did. So after we make our messenger RNA is going to leave the nucleus. And now we need to read the genetic code. And there's a couple of different types of charts that I will give you to read the genetic code. And we're going to read in a sequence of 3. So if we have a messenger RNA molecule AUGCCC TGT CAU. Let's start with that. We would take this chart right here and we'd have an A, a U, and a G, and that's going to code for methionine. So this right here is called our codon, and we're going to bring in, during translation, we're going to translate this code into an amino acid code. So our first one would be methionine. And then CCC, we'd get a C, C, and another C would be proline. So this codon would code for proline. I'm sorry, this wouldn't be a T. This would be a U. And so would this. This would be a U. UGU would be cysteine. And then CAU would be histidine. All right, and we would continue doing that, building our genetic code. Now, if you take a look at this other chart, if we start with AUG is our methionine, or our start. CCC, so we've got a C, we've got a C, and we've got another C. Sorry about that. Another C. This would be proline, and we put proline up here. UGU, we've got a U, a G, and then a U, that would be cysteine, and then CAU, is histidine. Alright, so we can use either chart, and I will give you both. Whichever makes more sense to you, you can use. Alright, now, there are 4 codons that are special. The first one we've already seen, AUG, is our start codon. Every gene, every new protein is going to start with AUG, and that codes for methionine. And then we have 3 stop codons, UAA, UAG, and UGA, and these are going to tell the ribosome when it can stop translating the code that is on messenger RNA. Alright, so in the nucleus, we went through transcription. I know these words are hard to read. So here's our nucleus. And in our nucleus, we went through transcription. So transcription happens in the nucleus. I don't know why it's erasing on me. But transcription happens in the nucleus. And then messenger RNA is going to come out. So here is our messenger RNA. Messenger RNA is going to come out, and it's going to find a ribosome. And at the ribosome, we are going to use a different type of RNA called transfer RNA. So this right here is our tRNA. That's a lowercase t, tRNA. And tRNA has a special shape. It has what we call an anticodon. So this is our anticodon, which is going to match up with our codon on our messenger RNA. This light blue thing right here, and this gold thing down on the bottom is our ribosome. Ribosomes have 2 different units, ribosome, a small unit and a large unit. And so the ribosome is going to connect to this messenger RNA, and it's going to bring in tRNA molecules that have the correct anticodon to match up with our messenger RNA codon. And it also has an amino acid. So this thing right here is an amino acid. And this one happens to be methionine because we have our start codon here. So we're going to start with methionine. And then we're going to bring in another tRNA that matches up with our, that has an anticodon that matches up with our codon, regular codon on a messenger RNA, and it's going to bring in the appropriate amino acid as well. So this is another amino acid, and we're going to connect these with a peptide bond. Peptide bond. After we've connected them, our first tRNA molecule is going to leave, and everything's going to shift over one spot. And we're going to bring in a new tRNA molecule with the correct anticodon to match up with our messenger RNA codon. And we're going to make another peptide bond. And everything is going to keep moving over. We're going to keep getting new tRNA molecules until we get to this one of our 3 stop codons. This is our stop codon. So our stop codon. And this is going to signal for our ribosome to let go of our messenger RNA. And it's going to kind of free this messenger RNA molecule so that we get free RNA nucleotides. And it's also going to release our protein. So as we've been linking these together over here, we've been building this peptide, or this polypeptide. Also known as a protein. And that's the point, because proteins are our worker molecules. DNA was just the cookbook that coded the information for the proteins, but these proteins, these polypeptides, are going to become amino acids and structural molecules that make us who we are. This is really the living molecule in genetics. DNA is kind of the code for genetics. So that's about it. We have transcription that happens in the nucleus, where we're making messenger RNA from our DNA template. And we're taking that messenger RNA out of the nucleus and connecting it to a ribosome and bringing in transfer RNA, tRNA molecules, with the correct anticodon and the corresponding amino acid, and we're connecting them together with peptide bonds until we make a polypeptide chain, which is going to be our worker molecule for proteins. That's about it for transcription and translation. I will see everybody in class.